Good morning, students. Today I am going to discuss session 3 of chapter 5 from your class 12 biology syllabus. The name of the chapter is Principles of Inheritance and Variation. Already I have made two sessions of chapter 5 from your class 12 biology syllabus, and from these two sessions we came to know about the genetics, some terminology about genetics, and the Mendel's monohybrid cross, their Punnett square, and the derived law. In this session we are going to discuss Mendel's dihybrid cross and the codominance and the blood group determination. So let's start the today's session. The first thing is the Mendel's dihybrid cross. Already we have discussed Mendel's monohybrid cross in our last session, and from this video I think we came to know that the Mendel's experimented two ways on the pea plant to determine the inheritance process, to determine the hereditary process. So already we have discussed Mendel's monohybrid cross. Today's topic is the Mendel's dihybrid cross. So before starting the today's class, you must know one thing first, that what is the basic meaning of the Mendel's dihybrid cross. Here the word is di. Here the word is di. Di means the two. Di means the two. Mono means the one and di means the two. So Mendel's dihybrid cross means in this experiment Mendel picked up two characteristics, two contrasting traits of the pea plant for his experiment. So actually, to verify his results of the monohybrid cross, Mendel also crossed pea plants differing in two characteristics. That's why this is also called the Mendel's dihybrid cross. So normally this helped, actually through this dihybrid cross, this helped to understand the inheritance of two genes at a same time. Actually he understood, actually he discovered the inheritance process of two genes, two pair alleles. So what is the basic definition of the Mendel's dihybrid cross? How you will define the Mendel's dihybrid cross? That the cross between two parents which differs in two pairs of contrasting characters. In the Mendel's monohybrid cross, actually he picked only one contrasting characters, only one contrasting traits for determining the inheritance pattern. But in this case, actually he crossed between two parents which differs in two pairs of contrasting characteristics. So this is the Mendel's dihybrid cross. Actually, after this experiment, it was found that inheritance of one pair of allele actually does not interfere in the interference of the other pair of allele. So what is the meaning of this? So when during the time of the gamete formation, when parents are giving 50% and 50% chromosome, gene, DNA, allele respectively, the father body gives 50% and the mother body gives 50%. But in this case what happened? That the two characteristics are not blending, two characteristics are not mixing. Okay, so this is the basic things, that inheritance of one pair of allele, it means the one trait, does not interfere in the inheritance of other pair of allele. So this is the basic interference, this is the basic conclusion of the Mendel's dihybrid cross. Next thing is the dihybrid cross. Actually this is the cross, this is actually the Punnett square. The parents, the P generation, here actually he picked round yellow and wrinkled green seed. So actually he picked two characters, one is the seed color, another one is the seed shape. I am repeating again, actually in this experiment he picked two characters of the pea plant. The first one is the seed color and the second one is the seed shape. So there are two contrasting color is there, that the yellow and green, and the two contrasting shape is there, two contrasting traits are there, that the yellow and the round and the wrinkled. Okay, so this RRYY and the smaller small y. Okay, this is the dominant and this is the recessive. So that's why this dominant actually always denoted by the capital letter, okay, and the recessive always denoted by the small letter. So from this capital R, capital Y, capital Y, we are getting RY in the form of the gametes, and from the smaller small y, we are getting small y in the form of the gametes, and we are getting in the F1 generation we crossed these two gametes, that the RRYY, capital R, capital Y, small y. This is the round YOLO. Okay, so this is the basic things of the dihybrid cross. So this is the Punnett square of the dihybrid cross. The first thing is this is the P generation. Okay, in the P generation we are getting capital R, capital Y, and smaller small y. This is the dominant and this is the recessive. This is this small capital R, capital Y is the dominant. Why? Because it's denoted by the capital letter, and smaller small y is means it is the recessive. Why? Because this is the recessive character. Why? Because the recessive character is always denoted by the small letter. Okay, now we are getting two gametes, capital R, capital Y, and the smaller small y. Okay, now we crossed the whole thing in the F2 generation. F2 generation means the F1 into F1. F2 generation means the F1 into F1. Now it means this is the F1 generation. This capital R, capital Y, small y. This is the F1 generation. So to getting the F2 generation, we have to cross F1 into F1. So it means capital R, capital Y, small y into capital R, capital Y, small y. Okay. After all the cross, we are getting total 16 individuals. So out of 16 individuals, 9 is round YOLO. Okay. Just count it. 1, 2, 3, 4, 5, 6, 7, 8, 9. So this 9 is the round YOLO. Okay. And 3 is the green round. This 1, 2, 3. Okay. Next is the 3 is the yellow wrinkled. 3 is the yellow wrinkled. That 1, 2 and 3. And the last one, only 1 is the green wrinkled. This is 1. So this is the basic thing. This is the basic phenotype of the Mendel's dihybrid cross. Okay. After that, after the whole procedure, so the Mendel actually derived one law. The Mendel actually derived one law. The name of the law is law of independent assortment. Okay. So for the Mendel's monohybrid cross, we are getting two laws. Okay. The first one is the law of dominance and the second law is law of segregation. And for the Mendel's dihybrid cross, we are getting one law only, that the law of independent assortment. And this is very important for your exam also. This is very important for your exam also, that these different kinds of the law. Actually, according to this law, what's actually this law states what? This law states that when two pairs of traits actually are combined together, combined in hybrid, so segregation of one pair of characters is dependent of the other pair of the characters at the same time of the gamete formation. So I am repeating again. When two pairs of traits are combined in a hybrid condition, then the segregation of one pair of characters is dependent of the other pair of characters at the same time of gamete formation. So all the characters are not blending together. All the characters are not blending together. All the characters are segregated separately, and actually the segregation, during the time of segregation, one pair of characters is dependent of the other pair of characters at the same time of gamete formation. So it also gets actually randomly rearranged in the offspring, producing both parental and new combinations of the characters. So this is the actually this law of independent assortment actually gives the variation to the new offspring. According to this law, actually this procedure provides the variation to the new offspring. Okay, so this is the inheritance pattern. Actually this is the inheritance pattern, this is the hereditary pattern. So I am actually repeating again that this law actually, so what is the main basic fundamental features of this law? That actually during the time of the gamete formation, the two pairs of traits when combined to form a hybrid, that during the time of the segregation, one pair of characteristics is independent of the other pair of the characteristics. So they are not dependent with each other. Okay, so this is the basic things of this law. This is the basic fundamental features of this law. Okay, next thing is the incomplete dominance. So what is the meaning of the incomplete dominance? Actually, all the patterns of inheritance would not be explained exclusively on the basis of Mendel's original principles. Okay, so what is the meaning of Mendel's original principle? That actually he derived three laws, that the law of dominance, law of segregation, and the law of independent assortment. But all the inheritance pattern, all the hereditary pattern actually cannot be explained exclusively on the basis of these three law. Okay, so later after so many experiment, so he discovered that 2 to 3 another things also. The first thing is the incomplete dominance. So what is the meaning of the incomplete dominance? Actually it's a phenomenon. It is actually one incident, it's actually one phenomenon where the phenotype of the F1 hybrid offspring does not resemble any of the parent. So I am showing the picture. So here the phenotype of the F1 offspring. Suppose this is the phenotype. Okay, this is the phenotype. What is the phenotype? That the round yellow. This is the phenotype, na? This is that F1 generation's phenotype is the round yellow. But they are resemble with the parents. There is another round yellow is there. So it's resemble with one parent. But sometimes it's happened that the F1 hybrid, this F1 offspring does not resemble any of the parent, but it's an intermediate between the expression of two alleles in their homozygous state. So what is the meaning of this? That actually scientist Corens discovered incomplete dominance in the Mirabilis jalapa flower. Okay, this is one plant, this is one flower. Okay, so it's also called the partial dominance, the semi-dominance. So what happened in the procedure of this incomplete dominance? That in this inheritance in which allele for a specific character is not completely dominant over other allele. Normally what happened? The Mendel experimented, Mendel discovered that one allele, particularly for one character, so one gene has two allele. Okay, so one allele is masked of the other allele. So that's actually carries the dominant gene, that actually carries the recessive gene. But sometimes what happened, that in which allele for a specific character is not completely dominant over the other allele. That's called the incomplete dominance. So the example is like the snapdragon or the Antherinum species, the cross between the two breed, true breed red flower and the white flower in the F1 generation is giving the pink color. Okay, and after the cell pollination in F2 generation, what happened? That the 1:2:1 we are getting this ratio 1:2:1. 1 means the red, 2 means the pink, and the another 1 means the white. Okay, so this is the basic thing, that the genotype ratio same as the Mendel's cross, but the phenotype ratio different than the Mendelian cross. So here these two, this capital or smaller, actually is not resemble with any of the parent, so that's why this gives the pink color. Okay, so this is the example of the Mendel incomplete dominance. So just look at the structure. This is the red color, this is the white color, but we are getting pink color flower. We are getting pink color flower, and the phenotype is capital or smaller. Okay, so that's why 1:2:1. Okay, that's why the ratio is the 1:2:1, because this is one parent, this is another parent, and the offspring is the pink color, and the name of the flower is the Mirabilis jalapa. And after that, after the crossing of these two flower, so we are getting one pink color flower. Okay, so this pink color is not resemble any of the parent, either it's not red or it's white. So this is the incomplete dominance. So actually I have made one cross also, that this is the red and this is the white, that capital R and smaller, then we are getting the capital R smaller gamete, and this is the pink color. This is the pink color, and we are getting the F1 generation. We are getting this in F1 generation, and after that, in the F2 generation means the F1 into F1. So when we are crossing capital R smaller into capital R smaller, then we are getting one capital R pure red, one smaller pure white, but two capital R smaller. Okay, so this color is the pink color. So this is the basic thing of the incomplete dominance. The same thing I have given, actually I have drawn another time, that this is the parent, that the capital R and capital W, this is white color, and in the F1 generation we are getting capital R W. Okay, but in the F2 generation what happened, that the one pure red, another pure white, but this two is the pink. So that's why the phenotype ratio is the 1:2:1 also, and the genotype ratio is also 1:2:1. Why? Because in the Mendel's experiment, what happened in the monohybrid cross, the phenotypic ratio is the 3:1, because it phenotypically it looks like tall. Okay, but in the incomplete dominance, what happened, phenotypically it also looks like pink color, it's not looks like red color or white color. So that's why the phenotypic ratio is also 1:2:1. And this is very important thing, this thing is very important thing, the phenotypic ratio is also 1:2:1, and the genotypic ratio is also 1:2:1. So this type of question may come, that in which dominance, in which thing the phenotypic and genotypic ratio is equal. So you have to write the incomplete dominance, you have to write this answer, the answer will be the incomplete dominance. Okay, next thing is the codominance. So what happened in the codominance? Here both the alleles for a character are dominant and express its full character is called the codominance. Actually this is one kind of the phenomenon in which two alleles actually express themselves independently when present together in an organism. Actually, so both alleles actually cannot mask up any of the characteristics. There is no dominant, there is no recessive, so both are dominant and express its full character. So that's called the codominance. Next thing is the example is the ABO blood group system of the human being. So normally this blood group in humans are controlled by the three alleles of a gene I. So actually this blood group is determined by the gene, actually the ABO blood group system are controlled by the gene I. Okay, so the plasma membrane, actually the plasma membrane of the RBC has the sugar polymer which actually protrude from its surface, and the kind of sugar is controlled by this gene. Okay, so this gene has total 3 alleles, not the 2 alleles. So they are the IA, IB and the I. Okay, I am repeating again, this gene has 3 alleles, IA, IB and I. Okay, and these alleles, this IA and IB, actually produce a slightly different form of the sugar, while allele I actually does not produce any sugar. So this I actually produce nothing, this I does not produce any kinds of sugar, but this IA and IB actually produce different kinds of the sugar. This ABO locus is located on the chromosome 9. So I think all of you understood properly what is the meaning of the locus. The locus is actually the home of the gene, the location of the gene. So already we have discussed in our session 1 about the locus. So this ABO gene, this I gene actually located on the chromosome 9. Okay, and IA is responsible for the production of the antigen A, IB is responsible for the production of antigen B, and I does not produce any antigen. It means I does not produce any of the sugars. So this is actually the codominance, the blood group. So we have total 4 blood group, the A blood group, B blood group, AB blood group, and the O blood group. So you have to remember this actually the chart, because from this portion many more questions may come. Okay, so A blood group means the phenotype of the A blood group is the IA or IAI. Okay, B blood group genotype is the IB or IBI. Okay, AB blood group means the IA and IB, and O blood group means only IO. It means the II. Clear? So this is the crossing of the blood group. Okay, suppose my father blood group is A and my mother blood group is A also. So what will be the offspring's blood group? So if you cross in this way, so this is the crossing of this A into A. It means the IA into IIA, so A blood group. IIA into IAIO, A blood group, and IAI and IAI, it means the A and O blood group. So there will be the possibility either A blood group or O blood group. Okay, suppose my father blood group is B and my mother blood group is B also. Okay, so what will be the offspring's blood group? That this is the cross, that IBIB into IBIB, it means the B. IBIB into IBI, it means the B. IBI into IBI, it means the B and O. So what will be the possible offspring blood group? Either B or O. Next thing is the, suppose the mother blood group is AB and father blood group is also AB. Okay, so what will be the possible offspring's blood group? That IAIB into IAIB, so it means the AB or A or B. And suppose mother blood group is O and father blood group is also O. So what will be the offspring's possible blood group? That the O. So you have to remember this chart properly, because this type of question may come, that in one parents the mother blood group is AB and father blood group is B. So what will be the possible offspring's blood group? Okay, so this type of question may come in a NEET exam and your other school exam also. So actually this is also called the multiple alleles. This is also called the multiple alleles. Why? Because this gene has 3 alleles. This gene has 3 alleles. So ABO blood grouping is also called the multiple allele. So what is the meaning of the multiple allele? So when more than 2 alternative forms of a gene occupy at the same locus on a chromosome. I am repeating again, when more than 2 alternative forms, it means the when more than 2 alleles of a gene occupy the same locus. So here the locus is same. I am showing here the locus is the same. Here the location is same, the chromosome 9. But this I gene actually occupy 3 alleles, IA, IB and I. So that's why it shows the multiple allelism. So the ABO blood grouping is a good example of the multiple allele. Okay. So actually 3 alleles governs the same character. These 3 alleles actually governs the same character, and the character is the blood group. The multiple allele is found when population studies are made that the single gene product may produce more than one effect. Okay, like the starch synthesis in the pea seeds is actually controlled by a gene having 2 alleles, capital B, small b. Next is the start synthesis effective if homozygous capital B and produce large start grade. Homozygous small b actually the lesser efficiency in the start synthesis and seeds are wrinkled. And the heterozygous capital B small b is round seeds and the intermediate size. Okay, so the most optimum thing is the when the seed is the gene is the homozygous capital B when the alleles are present within the start grain, this capital B, so it will be more favorable for the farmers. Okay, so actually this allelism, this multiple allelism, in this case, this ABO blood grouping case, more than 2, the 3 alleles are present which govern the same characteristics. Already I have told you that in the case of the multiple allelism, what happened, that the 3 alleles are present and govern the same characters. The same characters means this ABO blood group actually determine the blood group only. Okay, so the multiple alleles can be found only when the population studies are made. Okay, so this is the basic things of the multiple allelism. And the last topic for today, that the pleiotropy. Okay, pleiotropy and the polygenic traits. So there are another terms is there. So this is important for your 1 to 2 marks question. Okay, so it's actually the phenomenon in which a single gene product may actually produce multiple or more than one phenotypic effect. So what is the meaning of this? That when a single gene product may produce multiple or more than one phenotypic effect, not the genotypic effect. The main point is this is the phenotypic effect. Like one example is there, phenylketonuria. Okay, so this is one kind of the disorder, this is one kind of the disease which is caused by the mutation in the gene coding the enzyme phenylalanine hydroxylase. Okay, so these affect the individual, so hair and skin pigmentation and mental retardation. So actually it's showing 3 types of phenotypic effect. The first one is it shows the hair and skin pigmentation and the mental retardation also. So all those are different kinds of the phenotypic effect, not the genotypic effect. So you can show the skin pigmentation, you can see the hair fall, you can see the mental retardation. So all those things are the phenotypic effect. And the last one is the polygenic traits. So this is actually given the scientist Galton in the year of 1833. They discovered these polygenic traits. What happened in the polygenic traits? So this is actually in this way, in this term, the traits are controlled by 3 or more genes. Traits, any kinds of characteristics, any kinds of traits are controlled by 3 or more genes. That's called the multiple genes. So these traits are called the polygenic traits. So this is not the multiple allele. So in the ABO blood group system, actually gene is 1 but the allele is 3. But in this case, this is the polygenic. The poly means the many. So in this case, one trait actually controlled by 3 or more genes, not the allele. So this is the main important thing. This is the main basic difference between the polygenic traits and the multiple allelism. In the multiple allelism, what happened? That gene is 1 but the allele more than 2. Okay, but in the polygenic traits, what happened? That one traits are controlled by 3 or more genes. So this is the basic things of the polygenic traits. Like the one example is the human skin color. Actually this human skin color actually controlled by and caused by the melanin pigmentation. So there is one chemical pigment is there, melanin is actually present in the skin, and this melanin actually secreted this formed by the hormone melatonin. Okay, so that quantity of melanin is actually formed due to 3 pairs of polygenes like the A, B and C. So this A gene, B gene and C gene, actually all those 3 genes are control this melanin formation, the melanin pigmentation. Okay, so that's why the trait is the skin color. The human skin color is one characters, but these are controlled by 3 or more genes. So this is the another thing of the example. This is one actually basic example of the polygenic traits. So no more today. In our next session, we will discuss the another topic. I hope this video will help you a lot for better understanding, and please go through each and every session, each and every videos properly. And from this video actually we came to know the Mendel's dihybrid cross, incomplete dominance, multiple allelism, and the pleiotropy and polygenic traits. So please go through this properly, otherwise you will face problem during the time of your exam. Okay, so go through it properly and go through it thoroughly, and if you have any queries, ask me immediately. Okay, thank you, bye.